All right, folks online, can you tell me if you can hear me and see our slide? Perfect. Thanks, guys.
And this just goes.
Yeah. Okay, we're going to go ahead and get started with our next speaker. If you have attended our CEs for a while, Sally has kind of been a fixture for us, which is wonderful. She's always reliable to come and talk. She is a CDT and has been in emergency and specialty and primary care and for IDEX for a time, right?
Field.
Yeah, so laboratory, all the things. She has a wide breadth of knowledge and is always a reliable person for me to have come to our conference. So I so appreciate that. And yeah, we'll let you take it away.
I'm a noted lab rat. I really love all of the laboratory sciences just because there's so much that laboratory work can tell us that the animals can't. So they can't tell us they don't necessarily feel good or this hurts or they feel weak. So there's so much to be, in diagnostic laboratory. So I chose to go over urinalysis today just because I got a bunch of feedback from people saying they wanted just kind of an overview of urinalysis. So I can't really see people online, but in this audience, how many people actively do urinalysis on a day by day? Wow, that's awesome. Feeling pretty comfortable with them? No, for the most part. Anybody not and afraid of it? Well, that's good. That's unusual, but that's wonderful. So I will just go ahead and go on with the presentation. You said the arrow?
Yes. Weird, but here.
Oh, there we go. So I won't touch it.
Yeah.
Just the buttons.
Just the buttons.
Okay. So just a basic urinalysis overview, common laboratory diagnostic that gives the physical and chemical properties of the urine and is going to give an overall health assessment of the kidney and the bladder and the urinary tract system. It can also alert to other problems in organ systems. And it's essential for diagnosing a bunch of different metabolic diseases such as diabetes. It's a very valuable test in both healthy and sick animals to establish kind of a baseline when they're healthy and then something you can refer to as they age. You'll see changes with especially BU and creatinine with renal aging and injury. So going over the collection methods, the four main ones are cystocentesis, catheterization, free catch, and off of the floor, which we never like off the floor. But cystocentesis is where a sterile needle and syringe are used to collect urine from the bladder going through the abdominal wall of the animal. This is a primary gold standard for collecting urine because you're getting it in the most uncontaminated prospect. Also, anytime that you're sending anything in for urine culture and sensitivity, you're going to want to make sure that you try to get the cystocentesis because that's going to be your most accurate sample. Catheterization is a nice second unless it's a female dog or a female cat. That's not really great. But just knowing that it's a possibility. So using a commonly red rubber catheter, preferably the silicone. Silicone, kind of like what Foley catheters are. They've got a little bit more lubricity to the silicone. But red rubber catheters are a second best. I know that when I started way back in 1904, I think it was, we were using those PVCs. They came in these big long pipes and they were like really, really rigid. And man, I couldn't imagine being the recipient to one of those. So really try to be gentle and use a silicone or a red rubber catheter. They do have stylets that you can use within them, but again, making sure that you're very careful just because there's little fenestrations at the end of the catheter. And if that stylet of the catheter is too long, it can do some damage. So you want to be careful with that. And then we've got the midstream voiding, which is really, really common, especially in general practice. And then just getting any kind of baseline samples. Again, trying to make sure that you're getting kind of a midstream catch. Same, and I didn't mention that back for the catheter. With the catheterization, it's really super important when you're getting that initial sample that you're going to take 2 sterile syringes and expel, depending on the size animal. If it's a cat, like think about what your sample size is going to be. So if you think the most you're going to get is going to be 10 mils, you want to make sure that you just like discard the first 3 mils, because upon catheterization, you're going to have a lot of epithelial cells and debris and whatever else is lining that tract. So it's going to minimize the contamination from that urethra in the sample. So I wanted to make sure I went back into that. Same thing like with the midstream voiding, make sure that a little bit of the urine can come out and then start to grab your sample from there. Again, it's going to expel any kind of in intact male dogs, like smegma, sperm, anything that's been sitting in that tract for a while. So let them expel a little bit of that, and then you're going to get a much healthier sample. Something that's very commonly done with clients that'll catch it at home and then bring it in. They've got all kinds of different methods of doing it. They've got these big long sticks with little hoops that you can put things in. We used to use soup ladles at the clinic. There's a few different ways, but yeah, just making sure that it's mid-sample is best. And then the last but not least is off of the floor. And this is like really the absolute worst way of getting it, but sometimes that's the only thing that you can get. One thing that is common is, aside from the contaminants of whatever's on the floor, you've got to take into consideration any of the cleaning agents that were used on the floor. And they can absolutely transform the sample, so they can throw off all kinds of different tests. So the very most important thing with urine is that you want to observe it and do all the evaluation that you can within a 30-minute period of time. After that, it's going to start changing. So if you're collecting a sample, I always like to collect the sample, and I always save 2 tubes. So one, I always prepare that a culture and sensitivity is going to be done. There's nothing worse than having a pet come in and then you collect a urine, comes back and it's infection, and you don't have anything to send into the diagnostic lab for culture and sensitivity. So no matter what, I always make sure I separate those 2 samples. Plus, if the one that you're working on gets spilled or gets tipped over or whatever, you've got a backup. But again, just making sure that you're analyzing that within 30 minutes. If the clinic's really super, super busy, don't leave it on the counter. Make sure you throw it in the refrigerator and then go back to it when you have that 5 minutes to analyze it. If it's not examined soon as possible after collection, again, artifacts are going to continue to occur. Cells will lyse, crystals will form, changes in the pH. There's a multitude of things that will go on as time goes by. So yeah, just refrigerate it immediately. And then if it is refrigerated, make sure that you're always bringing it to room temperature. I usually say about 15 or 20 minutes is about a good time, because then you're getting to it before that 30-minute time frame. And always wear gloves, especially if it's a lepto case or suspected. Some prolonged sample artifacts. Again, you're going to see increased urine pH, microbial contamination or pathogens. The cells and casts are going to decompose, degradation of the chemical analysis. So you're going to see changes in bilirubins and ketones. Even after 24 hours, cells are going to continue to lyse, and bacteria will proliferate. It's not recommended. There's a handful that say yes and a handful that say no, that say to use EDTA or formalin. We just, working in the labs, just did not see that. There are some urine collection tubes that you can use that has a boric acid as a ingredient inside the tube, but that's only to be used for cultures and sensitivity. You can't do a urinalysis off of that, so you're not going to be able to do microscopic or your chemical testing with that. And again, make sure that all the samples are brought to room temperature if it was refrigerated. And then two, artifacts are still going to be observed with storage. So even if it's been a half an hour, 40 minutes, couple hours, a day or two, you're going to notice a lot of changes within that. So trying to get it in, get your urinalysis done that you're going to be doing in-house within that 20-minute period of time is ideal. So again, calcium oxalate, magnesium ammoniate, phosphate crystals can develop over time, and it's just a normal process. But you can't always report that, because if it wasn't present in the fresh sample that you initially observed, then it's not reliable to report it as that. And then you have the patient come in, do another collection, and then observe it within that 20-minute time frame. Sample handling, like I said before, always grab that urine knowing that you could be doing a culture and sensitivity. Again, it's nice to have a backup. And then it keeps the one sample as sterile as possible, so it's not sitting open in a lab, air contaminants, etc. Store it in a red top tube or a urine tube, and have it as backup. If you don't use it, you can always throw it away. So urine cultures, you guys remember doing microbiology? How fun was that? Seriously, that was one of my favorite classes. But it's a gold standard for diagnosing UTIs. If you've got animals that have persistent urinary tract infections, it's imperative to send that culture out. Again, I would strongly recommend using the boric acid tubes. They're like a yellow marble or a yellow top tube that you can get. A lot of times the labs will give them to you. If they don't give them to you for free, you can pay a small nominal amount for getting, a package of tubes. But let the trained professionals at a lab do this. I know that there's many clinics that are trying to cut costs and save measures, and they have their own little incubator, and they'll get blood agar plates. They have the blood agar plates, and then they'll just put the urine on there in a willy-nilly way. A lot of times stuff like that is going to grow anything anyway. So instead of wasting the extra day, 24 to 36 hours for any kind of bacterial growth to happen, just put it in a tube and send it off to the lab, because they have all of the calibrated equipment, the colony forming, the special inoculating ones that are calibrated. So they're going to be the best ones to perform that. So here's a tube of the boric acid. It's a yellow top, but they also have a gray or yellow top marble tube. The appropriate volume is generally going to be 4 mils, just like it is with blood collection tubes. They'll have a minimum bar right there of where you want your sample to be. If you cannot get that kind of a sample on the next slide, you can use a different culture tip, or you can put it in a red top tube and send that off. The big thing that's important to know is that you want to make sure that you keep the transport tubes chilled. So you want to keep that in the refrigerator until you're going to send that off. And then make sure that in the winter time, you're not letting it freeze. It's a nightmare in the winter here. Or keeping a cool pack on it so that it's constantly staying cool and not heating up. So if a urine transport tube or the minimum volume is not available, again, you can use a red top tube, where they do have transport swabs where you can let that swab soak in the urine for a certain amount of time, and then put it in the culture transport tube. But again, really important to make sure that these are being kept cool while you're transporting them. In-house step-by-step of urine. You guys probably already know that's like the back of your hand, but we'll go through it anyway. So it's collecting your sample, whichever way that you're doing it, between cysto, catheter, free catch, midstream. Doing physical observation, chemical testing, centrifugation, doing a wet prep and a dry prep. Do you guys both do wet and dry preps, or? Yeah. And then microscopic evaluation, and then send samples to a diagnostic laboratory if that's what you are going to do. So the equipment and materials are going to be there. The centrifuge, a microscope, a refractometer, the urine test tubes, reagent dipsticks, stain plus or minus. How many of you guys use stain? Do you guys use the SETI stain or new methylene blue? Yeah. High quality glass slides. And for those of you that have seen really poor glass slides, I think you understand how important the really good glass slides are for urine, because everything looks like artifact. And then Kim wipes or an absorbent towel. So the physical appearance consists of the color, clarity, and odor. Normal urine is typically transparent. Yellow and amber, depending to how concentrated the urine is. So looking at that, the clarity, holding it up against like newspaper or a book. We had a little reference sheet that we used that was just consistent at our lab that had print of different size like that we could repetitively mark whether it was clear, cloudy, or turbid. And then odor, normal urine has a slight odor from the ammonia in it, but it can be even more pungent with other issues. So a bacterial infection, UTI, can be real stinky. Goats have a really bad urine. Intact male cats have really strong urine. But it can directly correlate to bacteria. So if you've got a really stinky pee, the chances are that there's white blood cells that are proliferating in there. Urine chemistry tips. A lot of people are using multi-sticks. There's a multitude of them out there. Just really important to know that like leukocytes and nitrates are not going to be reliable. They're just not for animals. Urobilinogen is another one of those ones, bilirubin. But urobilinogen, generally you'll see a touch of it, but you just really pretty much ignore that because it's not significant. Important to keep these guys capped, not letting excessive light or air into them because it will degrade the strips surprisingly very quickly. Even taking a brand new bottle and if the exposure to air and light is more than an hour long, it can start to degrade those strips. So if you're not using them very frequently in the clinic, again, very important to just close that up, pick one out and close it up right after. I went through a little bit of the urine chemistries. So the pH, protein, glucose, ketones, urobilinogen, and blood. Just making sure that you're aware that the color from the urine is going to interfere with the pads. So if you've got a very, very concentrated urine, or if you've got bilirubin in the urine, icteric, it's going to interfere with the pads and reading those. So sometimes it's better to just go ahead and centrifuge the sample down and use the supernatant to run the chemistry strips off of it. That's not going to change the chemistry within the urine, but like leukocytes it would, because anything that's cellular or sediment is going to be at the bottom of the tube. But generally it's not going to interfere with the pH, protein, glucose, ketones, bilinogen, but blood it will. Centrifuging urine, it is really, really important to make sure that you're not over spinning it. Not time-wise, but the RPMs. So you want to make sure that you're using a urine-specific centrifuge. I know one that's really common is a stat spin. IDEX has one, Berger, Ingelheim has one. Beckman's got some. Just making sure that you can set the RPMs to about 2,000 to 3,000. And then you only need to spend your sample for about 5 to 10 minutes. And then the supernatant is poured or pipetted off, leaving a plug at the bottom or a sediment plug at the bottom. When you're reconstituting that, I mean, I've seen some people go in with pipettes and be pretty aggressive and go and sucking it in and out, and all you're doing is destroying cells. Casts that are in there are super fragile, so you're just gonna bust those open. So if you are using a pipette, make sure that you're very careful, having some of the sediment and not letting air go in. You can do just a quick in and out immersion at the very tip, but don't let excessive air and bubbles go in there. Other people will just take their hands and they'll flick the tube. Some people will swirl it. Whatever you need to do to reconstitute it is great. I like to do the simple swirl just because that's how I was trained in the lab. But yeah, and then from there, you're going to move on to either doing a wet prep or dry prep. So the wet prep slide is just as it shows. I'm one of the technicians that I like to use stain because it's just easier for me. I can use Cetastain or a new methylene blue, just making sure that you're not contaminating the entire sample. So I've seen some people that'll take the sediment and then they put the stain right in there. And I just don't agree with that because you are always going to have bacteria in the stain, no matter how much you filter it or how clean it is. Once you open it, it's going to show bacteria. So I like to do the dual slide. So I'll put on one slide, I'll put just the plain urine sediment and then another drop of that. And then on the cover slip, I'll turn it upside down, put a small drop of stain, and then put that on top of the urine and let it spread. The one thing that's really important about this is if you look at like how this slide looks, is that is going to be your ideal spread of the urine, because if you have too much urine and too much stain, that cover glass is going to float on top of that layer, and then you're going to have multiple layers of urine to look through. And so you're going to be adjusting your fine focus up and then down and then up and then down to read through layers and layers of the urine. So if you can get it so that the urine is just spread out to the edge of the slide, that's going to be your best and your fastest viewing space for looking at that. Dry prep slides are great for animals that can't pee and you have like 3 drops. What you're going to do is use it and treat it kind of like a blood smear. So you're going to put a drop on the slide, bring another spreader slide, back it up into the urine, and then spread it across. But instead of following through to get a feathered edge like you do with a hematology slide, you're going to do an abrupt lift about, I don't know, 3/4 of the drip through, however much sample you have. Because what that's going to do is going to give you a line at the end. So you're going to be able to see stuff towards the beginning of the slide, but towards the end, it's just like when you're sweeping a floor, all of your cells, all the big cells and important stuff is going to be down on that line edge. So you can get a quantitative overview of what's going to be in that slide. So if you're seeing bacteria, if you're seeing any kind of blood cells, casts or crystals, they're going to be pushed down to the end. So if you have a sample that you just really can't get enough, that's a really great way of doing it. I know a lot of other technicians and veterinarians that do like the dry slide, because although it's not quantitative, it's qualitative. So you're really going to be able to see a concentrate of what the cells are in there. So if there's not a whole lot to see on the urine and you want to do a dry prep to just really see if there's any kind of pathological or crystals or whatever, I really like doing it that way. Plus you can go ahead and use allow it to dry and use one of the Romanowski stains, like Dip Quick or whatever. And then you can view it microscopically. At that time it's dry, so you're not on a wet prep. You're not limited to just 10 power and 40 power or low and high dry. And you can go down to an oil power and see. That's what I like to do for bacteria. And then in that case, when it's bacteria and I'm trying to differentiate between the bacteria, the only way that you can really competently do that is with gram staining. So making sure that you're doing the gram stain and differentiating between gram positive and gram negative. So microscopic evaluation with a wet prep, like I was just saying, you're gonna start on 10 power. You're gonna be able to see those casts that are generally a little bit bigger, and you can identify them. A lot of times the way that those goes, they're gonna hang out on the outsides of the slide. So probably, I don't know, 2 to 3 fields in is the area that I will start with on 10 power. Scan for any kind of cast, any kind of crystals, and then move through to the inward of the slide. On 40 power, you're going to be able to get a magnification of those casts and be able to differentiate if they're red cell casts, white cell casts, hyaline casts, etc. Any kind of cells that you're seeing, white cells, red cells, leukocytes, sperm, fungi, and all the others. Fun stuff that you'll see in there. So just a brief run through of the urinary sediment. So the common things that you're going to be looking for are going to be your red blood cells, your white blood cells, the epithelial cells, which are broken down into renal, tubular, and squamous cells, your casts, crystals, and then any kind of infectious organisms, most commonly the bacteria. So red blood cells could vary in appearance depending on the tonicity of the sample. So getting used to understanding what crenated cells, or some people call them ghost cells, or in the hypertonicity of a cell, the red cell when it blows up is a ghost cell. But being able to recognize and knowing the difference between what is a white cell and what is a red cell. It's not really going to differ a whole lot size-wise microscopically than what you're seeing on a hematology slide, because they're the same. You're looking at them at the same magnification. But then just being able to, whether it's stain or not, be able to know that this is a white cell and these are red. And then down here, you can see the crenation with the hypertonicity. Yeah. And then white blood cells, to me, they just look glittery because they've got that stipulation that is the refractile of the cellular components in a white cell. So the red cells generally are not going to have them. The red cells can have a little bit of a halo effect, but the white cells are going to have that stippling. And then on that far slide, you can see where there's tiny bacteria. But I think it's really imperative when you're looking at slides to just get used to the environment that you're looking at, because you're going to be able to start noticing the size of the red blood cells, size of white cells, what the bacteria is looking like, and it'll increase your efficiency when you start doing them more and more and being able to go through them quickly. So epithelial cells, again, it's differentiating between those. The renal, tubular, and transitional ones are the ones that are not commonly seen, and that are more so ones that are indicators that there's some things and changes that are going on in the kidneys. So you want to make sure that you are paying attention to those. So you can see down at the bottom there, and at the pictures that are up there, the renal tubular cells are going to be distinguished by their centrally placed nucleus, where the transitional cells are smaller and rounder than the transitional epithelium, and the nucleus is going to occupy a little bit more of that cell. Again, when you start seeing these, you're more likely going to see them throughout the slide. So looking for different transitional cells to kind of hold them up against each other to help determine them. But the big key is knowing that they're not normal. And a sample like this, you would want to send out to a diagnostic lab and have a pathologist look at that. And we've got some squamous epithelial cells that you see very frequently, especially with the catheterized samples. And then we have some neoplasia going on. Of course, these are stained, but even on an unstained slide, you're going to be able to see those very large cells and the mitotic stippling that's going to be mitotic features that are going to be in there. Here's some examples of casts. I think red cell, white cell, and granular casts are going to be the ones most commonly seen for me. Hyaline casts you can see, but a lot of times by the time they're super fragile, they're glassy, they're not really diagnostic for anything, they're just there. Break and they're very clear. So I think the most common that I've seen are the red cell, white cell, and granular. You can see broadcast and waxy cast as well. My big recommendation is just make sure that you've got a lot of really good reference books in the lab, know what's not normal, and send it off. So crystal formation is directly affected by the urine pH and the temperature and the concentration. Also with the age of a sample. So if you've got a sample that's sat in a refrigerator for a while, more crystals are most likely going to multiply. There are some crystals that are classified also dependent on their color. So bilirubin, cysteine, tyrosine are crystals that are going to be on a yellow or brown spectrum. But again, they're always going to be identified by their shape and the urine pH because they're dependent on what that pH is that they're growing in. So the struvite triple phosphate, your typical plugged cat. You've got the coffin shape, refractile coffin lids. They're usually large and can be readily observed, but you can see little teeny tiny fragments of them all over the place. They can be found in urine of any kind, any pH, but they are favored in neutral to alkaline urine. The cysteine crystals, these are the rectangular ones. I think that they kind of have a little bit of color to them, but I maybe have just looked at them too long. But they're the rectangle and hexagon type. They can be small to medium and large. Sometimes you'll see them in clusters, or they kind of will layer on top of each other and stack. But they really are favored in acidic urine. Cholesterol crystals. They look like little underwater cities to me. Again, they're likely to appear in urine sample that's been refrigerated. So making sure that you're letting that temperature come to, or the sample come to room temperature. You're going to find them in both acidic and neutral urine. You don't really see these very commonly in animals, but you can, and they're not super significant, but you always want to report them if you see them. So your tyrosine, these look like little haystacks. These are going to be brown in color or dark in color. They're often found in acidic urine and are caused by a lot of metabolic disorders like liver disease or tyrosemia. And the bilirubin, you guys get excited when you see bilirubin in the billy samples? Because they're fun and they're pretty and they're orange. They're just fun because they're really hard to not miss. So again, needle-like appearance, they're going to be yellow to red in color. Again, high levels of bilirubin or they don't have a preference in acidic or neutral urine there because their bilirubin is caused by the bilirubin that's in the urine. Calcium carbonate crystals, these are all variable in size and they look like those spheroids, like a cut tree. They can be colorless to a little bit of a yellow color or a brownish color. When they occur in high numbers. Again, this is not a real common crystal that I have seen. It's more the calcium oxalate that I've been exposed to, but another one of those examples of you'll be able to identify it and know that it's not normal and send that out. Here's your calcium oxalate that I was just describing as well. The dihydrate crystals typically are colorless in squares, kind of like little envelopes. They can both occur in neutral and alkaline and very frequently seen because of that. They can vary in size. Sometimes you'll notice that they'll cluster in one part of the urine slide. So making sure that you're scanning that whole slide side to side. You can see them on 10 and 40. In some cases, large numbers of the tiny oxalates can appear amorphic debris, where you're not actually seeing that envelope shape. They can break apart and they'll look like amorphous. So it's really important to go up to that high power magnification so that you can identify them. Ammonium biurate are another one of the crystals that is yellow to brown in color. And these crystals, they look like they have little spiky thorns, can resemble small bugs. They're often found in the alkaline urine, but can also be seen in normal urine, not very common. These we'll see an increase of in the lab because the sample has gotten old, like 2, 3, 4 days old. And they're fun, but they're not really diagnostic in-house. If you see these in-house on a fresh urine sample, there's a problem and the sample should go out. But if it's an old sample, like I said, we come and saw these in the lab on old samples. So leucines crystals are another one of those ones that are the yellow-brown color. You're going to find those in the acidic urine as well. They're not typically found in healthy urine. So when you do see these, there's usually a problem that's with the liver. So making sure that you're checking out all the liver chemistries and the fun stuff with liver disease as well. But the big thing about them is they are a little bit larger in size and they look like a cut tree trunk. Amorphous debris. So there's 3 or 4 different kinds. You can see it in acidic urine, you can see it in neutral urine, you can see it in, it looks like a fine refractile pile of powder, essentially. They can take on any kind of shape. They can be urates, phosphates, or xanthine. Again, they're small crystals. They're difficult because they look like artifact, but you can tell because there's little piles of them and they're going to be refractile. You're not going to be able to distinguish whether they're urates, phosphates, or xanthine by visualization. It's just important to make sure that you're reporting that, how much of them you're seeing. And letting them know that they are present in the urine. A lot of times people do confuse these with bacteria. The one way that I always showed or distinguished between the two is that the amorphous crystals are going to be refractile where bacteria is not. Bacteria will come kind of in and out of focus, but it doesn't refractile like the amorphous debris or amorphic crystals do. And then there's infectious organisms, which is primarily your bacteria. E. coli is the single most common pathogen for reoccurring UTIs. So it's going to be the most common bacteria that you'll see in those. So when you see, if you're looking on the slide and you're seeing bacteria on a non-stained wet prep or the dry prep, chances are if it's a rod, it's going to be your E. coli. And the other common pathogens include staph, Proteus, strep, Klebsiella, and Pseudomonas. So those are really your top bacteria with urinary tract infections. So here's some other pictures of bacteria. These are with Diffquig, some are with gram stain. Again, you're going to be able to see the real true identification of those bacteria, whether they're gram negative, gram positive, if they're spirochetes, if they're chenes, if they're clusters. But they have to be differentiated by gram stain. Do you guys all use gram stain in the clinic or no? No. Gram stain can get kind of pricey and it's really a long process. It's not a really quick staining process. But another reason to ship it off to the lab and let the laboratory geeks do their stuff. And then the last thing worth mentioning is your neoplasia. So transitional cell carcinoma is going to be absolutely the most common. A lot of times you're going to see those with blood cells in them because they're nasty tumors. But you're going to see these big, huge, massive cells that you're going to know it's not a squamous epithelial. It can, the medium-sized ones can be about the same size of a squamous epithelial, but you're going to see some really big ones. A lot of times you'll see dividing nuclei, and then you'll also notice mitotic figures in those under stain. And if you see them, tell the vet and send it off to the pathologist. So reference books. These are 4 of my favorite ones. My very, very favorite one is the one in the top middle, Carl Osborne, who is like the grandfather of urinalysis at the U of M, co-wrote that book, and there's amazing, amazing color plates in it. But any of those reference books are going to be great to have, especially for constituents in the urine that you don't see very often. You're going to have something that you can guess or play a game, you know, who's going to win, what it is. So yeah, I always like to have reference books. And then in summary, urinalysis is really something that can be done inexpensively in the clinic. There's a lot that you can do within that 20 minutes. And we're properly trained. And if you want more training, get more training or see more CE or have someone come into the clinic and do some more training with you. But there's so much that can be done in that first half hour before sending it off. Again, remembering with urine cultures to get some of those boric acid transport tubes, it's just going to really give you the best results for your contaminations. Physical, chemical, microscopical, again, it's all going to be helpful in doing any kind of diagnosing of disorders, diabetes, pre and post kidney disease, cancers, yeast infections, and all that stuff. So that's all I've got. Do you guys have any questions? Oh, there's a bunch of people that are mentioning the SediVue, the IDEX SediVue. Have any of you guys used that? Do you guys like it? Yeah. Yes. So go IDEX for that, huh? Oh, sorry. Looking at the chat.
Do you recommend a specific amount of urine to spend for your urinalysis?
It really depends on your on your patient, because like I said, if you've got a polyuric animal or someone that's got a UTI in there or peeing all the time, or they feel like they have to pee all the time, you just got to work with what you got to go with. For a normal healthy animal, I like to do about 5 to 10 mils from a dog. And again, before you're testing that, making sure that you're not shaking it, but that you're well reconstituting it if it's sat, because all those cells, crystals, and everything are going to start settling. So I always say probably 5 to 10 inversions. For a cat, good luck with whatever you get, if they've got a bladder. But yeah, I mean, there's times where all I've had to work with is like a hematocrit tube, and you're like, okay, well, we need the pH and we need specific gravity. And then I'm just gonna throw it on the slide and do a dry prep, because then whatever's left over, you've got. But that's a good question. Yeah. I don't know if anybody else has. Thank you to all who've listened. How do you get to the non-pointer?
Yeah.
I wasn't paying attention to the chat.
No problem.
Did I run over time or I'm good?
No, you're fine.
Okay. So if you can't look at it immediately, can you spin it and then refrigerate the sediment? Yes, you can. But what I would recommend in that case is if you can't look at it immediately, I would do a dry prep and then have the sediment and go back and do a wet prep, because you're gonna be able to see some changes within that. You'll see the original crystals in the fresh sample versus anything that could have grown with an old sample. But if you're doing it really truly within a refrigerated sample within a 12-hour period, you're not gonna see a huge amount of changes. It's just after that 12 to 24-hour period. Sweet.
Yeah, I think that's it.
All right.
All right.
Well, thank you, everybody. Have a good day and a happy Turkey Day.
Great. We're gonna take a break and we'll get started again at 2 o'clock.